Now we are entering into eukaryotes. Okay,
Yes, ma'am.
that is the last part of regulation of gene expression part 3. Okay.
Yes.
Okay, Sandhya. Then gene expression in eukaryotes, you can mute now. Okay. Gene expression in eukaryote. Each cell of a higher organism that contains the entire genome. Gene regulation of eukaryote is almost same as prokaryote. We know there are two types of gene, inducible genes and constitutive genes. Constitutive genes are known as housekeeping genes. So expression of housekeeping genes in most of the cell. Then activation of selected genes only upon demand occurs. Then there is a permanent inactivation of several genes. Okay. In prokaryotes, most of the DNA is organized into genes. That is the difference between prokaryotes. We are entering into the difference between prokaryotes and eukaryote gene expression. Okay. In prokaryotes, most of the DNA is organized into genes, but in mammals, very little of total DNA is organized into genes. Okay. The bulk of the extra DNA is not known. The function, there is an amount of bulk DNA. The function of that bulk DNA is not known. Okay. So that's why eukaryotic gene expression and its regulations are highly complex. That is the reason. In case of prokaryote, DNA contain active genes. Okay, but in case of eukaryote, DNA contain active genes along with bulk genes. Okay, the bulk DNA part. Okay, that is not organized into genes, and the function of that extra DNA is not known. Okay, Sandhya, you can unmute. Okay.
Yes, ma'am.
Okay, what is the difference between prokaryotic gene expression and eukaryotic gene expression?
In each cell of higher chromosome that contains entire genome, in eukaryotes, it is almost same as prokaryotes. Expression of housekeeping genes is in most genes, cells, sorry. There is in prokaryotes, most of the DNA is organized into genes, but in mammals, very little amount is in genes.
We already discussed that bulk DNA, name of that functionless DNA. Did you remember?
No, ma'am.
One special word is the
Junk DNA.
Okay, can be expressed as junk DNA. One term is there. You just revise. It is present in our gene expression part 1. Okay, inducible DNA, constitutive DNA, and then one part of DNA. That part of DNA is known as functionless DNA. When we express types of gene, we express which one is known as constitutory genes? Sandhya?
Yeah.
Constitutory gene, constitutive genes. Housekeeping genes.
Yes.
Why they are known as housekeeping genes?
It's because they transcribe. Gene is transcribed.
Okay, they are present in all time. Okay, yes. Every time they are there. Okay, yes. Genes are present. That's why they are known as housekeeping. They control all the activities inside the house. Okay. Okay. That's why they are known as housekeeping genes. Okay.
Yes.
Okay. Then, what is the main difference between eukaryotic and prokaryotic gene expression?
In this thing, what is that? Most of the DNA is into genes in prokaryotes, but very little amount of DNA is in genes in mammals.
Okay. Then next we are entering into chromatin structure. Okay, you can mute now. Chromatin structure and gene expression. The DNA in higher organism is extensively folded and packed into protein. DNA complex is called chromatin. Okay, chromatin is a protein DNA complex. In higher organism, that DNA is folded and packed into protein DNA complex. It is called chromatin. Okay, what is the role of chromatin? This chromatin structure provides an additional level of control of gene expression. Okay, it is not just packed or folded. It is folded and packed for a purpose. What is the purpose? That provides an additional level of control of gene expression. Okay, genes that are transcribed within the cell are less condensed. That point is very important. Genes that are transcribed within the cell are less condensed and more open in structure. In contrast, the genes that are not transcribed, which form highly condensed chromatin. So here we discuss 3 points. What is chromatin? Chromatin is a protein DNA complex that is extensively folded, and DNA that is extensively folded and packed into protein DNA complex. It is known as chromatin. What is the purpose of chromatin? That gives an additional level of control of gene expression. How it is controlled? There is a less packed area, less condensed area and more condensed area. Okay, genes that are transcribed within the cell are less condensed and open. Genes, in contrast, genes that are not transcribed, which forms highly condensed chromatin. Okay, you can see the chromosome. This is chromatin fiber, and we know the histones, beads on string model, DNA bound on nucleosomes. Okay, these are the structure of chromatin. Okay, then I explain with 2 structure. Okay, then Sandhya, you can unmute. Hello. Hello, Sandhya. Yes. What is the role of chromatin?
Chromatin, it's an extensively folded and highly and packed into protein DNA. It's called chromatin. It provides an additional level of control of gene expression.
Okay. What is the difference between genes to be transcribed or not enter into transcription?
The genes that are transcribed are less condensed and more open in structure.
Then what about the genes are not transcribed?
They're highly condensed.
Highly condensed. Then what is the structure of chromatin? What is your idea about chromatin structure?
Many nucleosomes come, histone proteins around nucleosomes.
Okay, first of all, there are chromatin fibers, then histone proteins, then we can arrange it like a beads on a string, DNA bound on nucleosomes. Okay, yes. So actually, what is chromatin? It is a DNA protein complex. Okay.
Yes.
Okay. Okay, then next. Okay, you can mute now. Next, we are entering into a topic that is known as histone acetylation and deacetylation. Okay, we know the protein part is histone, histidine containing protein, histone. The heading of topic is histone acetylation and deacetylation. Eukaryotic DNA segments are wrapped around histone proteins to form nucleosome. Okay, then acetylation and deacetylation of histidine, that is an important factor in determining this gene expression. So chromatins formation is important for the control level of gene expression. How it is controlled? How this chromatin structure control the gene expression, that we are going to discuss. So eukaryotic DNA fragments are wrapped around histone proteins to form nucleosome. Then acetyl group is added or removed. Acetylation and deacetylation of histone is an important factor in determining the gene expression. Acetylation, that leads to, that is the most important part. Acetylation will lead to activation and deacetylation will lead to inactivation. Then in which part this acetylation occurs? We know histone is a protein that is made up of amino acid. So acetylation occurs on lysine residues in the amino terminal end of histone. Histone is a protein. Protein always write from N terminal to C terminal. So the N terminal contain one amino acid, lysine, and this acetylation occurs on lysine residues in the amino terminal end of histone. What happens? Histone is modified. The modification of histone that reduces the positive charges of terminal end and decreases their binding affinity to negatively charged DNA. Okay, we know when at the time of transcription, what happens? We need an open structure. So consequently, nucleosome structure is disrupted to allow transcription. Okay, that's why acetylation leads to activation and deacetylation leads to inactivation. You can see from the structure, you can see the beads on rings model. Histone proteins are there. Orange color that represents genes. So histone, when it is get acetylated, this one more thing is the methylation. The second diagram that expresses acetylation. So histone tail acetyl group is attached to histone. Histone acetylation resulted loose packing of nucleosome. We can see the tight packing is released. Okay, then it is converted to loose packing of nucleosome. Transcription factors then only can bind with DNA and genes are expressed. Okay, Sandhya, you can unmute.
Yes, ma'am.
Okay, Sandhya. Okay, what is acetylation and deacetylation of histone?
Acetylation, it is of histone. Acetylation leads to activation. Deacetylation leads to inactivation. Acetylation occurs on lysine residues in the amino terminal end of histones.
Which is the normal sequence of protein? For the synthesis of protein, we need a sequence from C-terminal to N-terminal or N-terminal to C-terminal.
N-terminal to C-terminal.
Okay, N-terminal to C-terminal. Okay, very good. Then continue.
Yes, ma'am. The histone modification reduces the positive charges on the terminal ends, and affinity for binding decreases because of negatively charged DNA.
Okay, then what happens? Affinity decreases, then what happens?
The nucleosome structure is disrupted, that will allow transcription.
Okay, tight bound nature that will cause us inactive. Okay,
yes.
You can see from this diagram what happens at the time of acetylation. This nucleosomes are closely attached. In the case of first diagram, you can see in the first diagram each part of histone is tightly attached. Yes.
Yes, ma'am.
Yes, after acetylation, this acetyl group is attached to lysine residues in the N-terminal of protein histone. What happens? Acetyl group is there, AC. Okay, so when this acetyl groups are attached, what happens? Activation of gene expression. What happens? The actual structure disruption occurs and they are far apart. There is a distance. Yes?
Yes, ma'am.
Then the genes are expressed. The hiding genes are removed. They are open now. Okay.
Yeah.
The first structure we can't find out. There is red color genes, but we can't find out. But in the second structure, we can easily find out. The transcription factors can bind on the gene. Okay.
Yes.
After acetylation. Okay. I think it is clear.
Yes,
Sandhya? Okay. Then next part, you can mute now. Next is methylation. Acetylation leads to activation. Okay, here the second part is methylation. Methylation of DNA that will leads to inactivation. Okay, so cytosine. There is cytosine, adenine, thymine, cytosine and guanine. They are the nucleotides in the DNA. So cytosine in the sequence of DNA get methylated and forms 5' methyl cytosine. Okay, a major portion of CG sequence 20%, almost 20 to 70% in human DNA exists in methylated form. Methylation that leads to loss of transcriptional activity and thus inactivation of genes. Okay, methylated DNA is not exposed and bound to transcription factors. Methylation DNA correlates with deacetylation of histones. Okay, you can see from this diagram. Okay. Here it is methyl group is the green color methyl group. Methylation of DNA and histones causes nucleosomes to pack tightly together. Okay, they are close together after methylation, like our deacetylation. So methylation of DNA and histones causes nucleosomes to pack tightly together and transcription factors cannot bind the DNA and genes are not expressed. Okay, Sandhya, unmute.
Yes,
hello. Okay, then what is methylation?
The It causes the nucleosomes to pack tightly together.
Yes, the basics of methylation, in which area that get methylated, in which nucleotide that methylation occurs. Huh?
Actually, ma'am, that slide little disturbance. I couldn't see that.
Okay, now you can see the slide.
Uh,
Yes, Sandhya. Can you
yes.
see the slide? Yes.
Yes.
Can I explain? You want explanation more?
Yeah, one more thing.
Okay. Cytosine. We know there are different nucleotides. Cytosine, thymine, okay, adenine, like that. Okay. So cytosine in the sequence of DNA that get methylated. Okay.
Yes.
And forms 5-methyl cytosine.
Yes.
Okay. So a major portion of cytosine CG sequences in human exist in methylated form. Okay.
Yes.
At all time, the genes should be in inactive. That when it is essential, then only it converted to active form, right?
Yes.
Otherwise, what happens? Unnecessary factors are always synthesized in our body, right?
Yeah. Yes.
Yeah. So a major portion of CG sequences in human DNA exist in methylated form. So methylation leads to loss of transcriptional activity and thus inactivation of genes.
Okay.
Then methylated DNA, then same as our deacetylation. We discuss our deacetylation. Okay.
Yes.
What happens in deacetylation?
The acetyl group is removed.
Acetyl group is removed. And what about the structure of nucleosomes then?
It becomes inactive.
It becomes inactive and tightly packed.
Yes.
Right?
Yes.
What happens in acetylation?
That causes activation. It occurs on lysine residues.
Okay. Then what happened to the structure? Tightly packed or loosely packed? Okay.
Yes, miss.
Loosely packed or tightly packed after acetylation? Sandhya?
After loosely packing, ma'am.
Okay. Then the same thing happens in case of methylation, like our acetylation, deacetylation. Methylated DNA is not exposed and bound to transcription factors. Okay.
Yes.
That methylation of DNA correlates with deacetylation of histone, same as deacetylation. Okay. Okay, you
Yes.
can see this structure. Can you see the structure?
Yes, ma'am.
Okay, that blue violet color. Okay, so you can see from this structure, methylation of DNA and histones causes nucleosomes to pack tightly together. Now it is tightly packed, right?
Yes, yes.
What happens in case of acetylation? It is loosely packed. We can easily find out the genes here.
Yes,
yes. This is same as deacetylation. In case this structure is same as that of methylation and deacetylation. You can see from the second diagram what happens in deacetylation. It is also closely packed. Yes. Then only in case of acetylation it is loosely packed. Okay.
Yes.
That structure, tightly packed structure disruption occurs after acetylation. Okay. Yes. I think it is clear now.
Yeah.
Yes. Yeah. Okay. Okay. Then you have a doubt only at the site of cytosine, right? Yeah. What happens at cytosine?
Methylation.
Okay. Occurs. In which site acetylation occurs? Lysine residue.
Lysine residue. Yes, ma'am.
Lysine is present in histone.
Histone.
Okay. Yes, then methylation that occurs on DNA. That is the difference.
Okay.
Yes, methylation occurs on DNA. Acetylation occurs on histone.
Yes.
Okay. Histones are made up of amino acid. A lysine amino acid present in the N terminal of histone. In that area, acetylation occurs.
Yes, ma'am.
Yes. Then what is the difference between methylation? That methylation occurs on DNA. In which part? Cytosine residues.
Yes, ma'am.
Now it is clear. I think that the confusion occurs in the methylation and acetylation area, right?
Yes. Yes, ma'am.
Okay. Then we already asked questions. Then acetylation of histone, methylation of DNA, and chromatin structure. Okay. We finish that topic. Any doubt?
No, ma'am. Now it's clear.
Okay. Then can I ask 3 questions?
Yes, ma'am. Yes, ma'am.
Okay. Acetylation occurs in which part?
In the lysine residues in the amino terminal ends of histones.
Very good. Then what is chromatin structure?
It's an additional level of control of gene expression.
Okay. Then the structure, how it is formed? Chromatin.
The chromosomes they condense, they form around histone proteins. They wrap around histone proteins like beads.
Okay. DNA coiled around histones.
Yes, ma'am.
Okay. That is very easy to remember our acetylation, methylation like that. Okay. DNA that is coiled, tightly coiled around histones.
Histones.
Okay. The different types of histones are there. Okay.
Yes.
Okay.
Yes, ma'am.
Okay then. So I think this part is clear. No problem.
No problem.
Okay. Okay. Okay, Sandhya, you can mute now. Then we discuss this enhancers in case of our transcription. So here we are going to discuss this detail about enhancers. Enhancers or activators of DNA elements that facilitates or enhance gene expression. Enhancers, they provide binding sites for specific protein that regulate transcription. They facilitate binding of transcription complexes to promoter regions. Tissue-specific gene expression is largely mediated through the involvement of enhancers. So there is a simple thing, enhancers or activators. We know there is a promoter site to which our RNAP binds, and or in case of eukaryotic transcription, transcription factors bind to that area. So what is the role of enhancer? Enhancer, they provide binding site for specific protein that regulate transcription. They facilitate the binding of the transcription complexes to promoter region. Then in each cases, there is tissue specific gene expression. It is largely mediated through the involvement of enhancers. Okay, you can see here there are DNA bending protein is there. Here there is a promoter site, and promoter site, you can see RNAP binds at this promoter site, transcription factors and mediator proteins are there, and enhancers bind and find out to this area. Enhancers or activators, you can see these 3 activators, they find out and helps for the attachment of transcription factors and mediator proteins to promoter site. Okay. Okay, that's about our enhancers. Okay, Sandhya, you can unmute.
Yes, ma'am.
Okay, what is your idea about enhancers? Explain.
Enhancers enhance the gene expression. They provide the binding site for a specific protein that helps in regulation of transcription. They facilitate the binding of transcription complexes to promoter regions.
Okay, they provide all requirements. Okay,
Yes.
that is simply we can say they provide light, food, and everything. Okay, enhancers provides everything for the binding of transcription factors, mediator proteins to the promoter site.
Yes.
Okay,
Ma'am.
simply
Yes.
that's all. Enhancers, enhancing mechanisms. Okay.
Yes. Ma'am.
Okay. Okay. Then we can enter into the next topic, motif.
Yes, ma'am.
Okay. Have you heard about motif?
No, ma'am.
Okay. Okay. It's a special area. It's a simple thing. In this case of genetics, so many words are there. So we stuck on those words. Motif. What is that? Then it is simple matter. Okay.
Yes.
So split each and every part. Then keen attention. Okay. Okay. Then you can mute now. Motif in proteins and gene expression. A motif literally means a dominant element. Motifs in protein that mediate same thing, binding of regulatory proteins or transcription factors to DNA. Okay, the specific control of transcription occurs by binding of regulatory protein with high affinity to the correct regions of DNA. A great majority of protein DNA interactions are brought out by 4 unique motifs. Okay, there are 4 motifs present in our DNA. Okay, for the transcription. Helix turn helix, zinc finger, leucine zipper, helix loop helix. Okay, once again, what is the role of motif? Motif means a dominant element. Then what is the role? Motif in proteins that mediate. In protein, generally it's a protein, it's a part of protein. Okay, then motif in protein that mediate the binding of regulatory proteins to DNA. So it's a highly controlled mechanism. Transcription is not an easy process. Now we understand it's a heavy heavy controlling mechanism is behind the transcription. The body take that much effort to synthesize each and every protein. Okay, so its specific control transcription occurs by binding of regulatory protein with high affinity to the correct regions of DNA. So for that, a great majority of protein DNA interactions are brought out by 4 unique motifs. That is helix turn helix, zinc finger, leucine zipper, helix loop helix. Okay. Okay, Sandhya, you can unmute.
Yes, ma'am.
Hello. What is motif now? Now you understood. What is motif?
The dominant element, ma'am.
Okay. Then what is their function?
They mediate the binding of regulatory proteins to DNA.
What is the regulatory proteins? What is meant by regulatory proteins? Transcription factors.
Transcription factors. Yes, ma'am.
Okay. In eukaryotic transcription, we discuss TF2A, TF2F, TF2H, like that. Okay. Okay. So it's a part of protein. Okay. It's not a single area, small part of protein. Okay,
Yes,
Sandhya. Okay. Then you can mute now. First one, that is helix turn helix motif, HTH. This word helix is very familiar in the case of secondary structure of protein. Okay, so helix turn helix motif. It's about 20 amino acid which represents a small part of large protein. HTH is the domain part of the protein which interacts with DNA. Example, in case of lactoperon and also in CAMP dependent cap of E. coli, we use this helix turn helix motif. Okay, then the second one is zinc finger motif, TF2A, that contains the zinc ion as a repeating coordinated complex. This complex is formed by the closely spaced amino acids cysteine and cysteine followed by histidine and histidine. The zinc finger bind to the major groove of DNA and lie on the face of the DNA. And in which cases we use the zinc finger motif? Steroid hormone receptor transcription factors use zinc finger motifs to bind DNA. Okay, Sandhya, you can unmute.
Yes, ma'am.
What is this helix? Have you heard about helix, alpha helix?
Yes, ma'am.
Yes, right-handed alpha helix and left-handed alpha helix. In which area we discuss this helix?
In proteins structure.
Which type of level of organization? For primary, secondary, tertiary or quaternary?
Secondary proteins, ma'am.
Okay, you can see this wound round like a spring. Okay, I can't draw. Okay, right-handed and left-handed helix is there. Okay, right-handed is very common. Okay, then it's about 20 amino acid. Here, this helix turn helix. I will show you the diagram.
Yes.
Okay, you can see the diagram. Yes, helix turn helix. Can you see?
Yes.
Yes, in the DNA we can see this helix turn helix. It is not alpha helix. The structure is helix turn helix. Okay.
Yes,
okay, 20 amino acids are there in case of helix turn helix. Okay, it's a small part of large protein. HTH is the domain part of the protein that interacts with DNA, and this motif is present in our lactoperon. Okay.
Yes.
Lactoperon, and one more thing is the CAMP dependent cap of E. coli. That is used in which condition?
Lactoperon.
Lactoperon, but one point is there.
When lactose is present.
When glucose and lactose, we provide glucose and lactose.
Glucose is exhausted.
Yeah, that's the important point. When glucose is completely exhausted, there is no glucose, then some sort of catabolite. Yes, that catabolite will produce catabolite activate protein.
Yes, ma'am.
Then only it will be converted to CAMP. Okay, that is the basic. It is then only lac-operon functions. Okay.
Yes, ma'am.
Okay, we must note that point. When glucose is exhausted, there is no available glucose is not present. Okay, no availability of glucose. Okay. Okay, then that's about helix turn helix. I think it is clear. Helix turn helix motif. Yeah.